Hello, this is Dr. White, and I'll be discussing about osmosis and also your lab. So, the learning objectives of this lab is to define concentration in terms of solute and solvent. Additionally, you're going to define osmosis in terms of concentration differences. Also, you are going to explain the direction of osmotic pressure for conditions of isotonicity, hypertonicity, and hypotonicity. You'll also be distinguishing the movement of solutes from movements of water in osmosis, as well as compute ratios of concentration differences between two neighboring solutions. And lastly, you're going to calculate concentrations of unknown solutions after testing osmotic pressure of known solutions. So, just to give you a quick review, your cell, especially animal cells, have cell membrane. Now, for plants, they have cell wall. Now, there are cellular compartments that is located along the boundaries of your cell membrane. This maintains water balance in and outside of the cell. So, just a quick introduction. We already know that macromolecules are involved in cell to cell interactions and interactions with the environment. Now, macromolecules serve many roles in living systems, and they can be very small, like the nucleic acids, or they can be very large, like proteins. And today we are going to look into how these molecules can get in and out of the cell, specifically in this case, water, which is related to the concept of osmosis. Now, in our cell membrane, there are specific protein channels that regulates the access of these substances to the intracellular space. The one role of the molecules is to protect the cell. Now, the intracellular compartment is a controlled environment. As you can see in this diagram here, there are some sort of channels that allows substances to go in and out of the cell. And this creates a concentration gradient between the intracellular, which is the inside space of the cell, and the extracellular space. Now, water is essential for all cells, and most of the water in the human body is in the intracellular compartment. And water can freely move in and out of the intracellular space through specialized channels. And as soon as the amount of water is equal to the amount of the inside and the outside of the cell, a balance is achieved through osmosis. Now, in the concept of osmosis, too much water leaving the intracellular compartments can cause cells to shrink, and you also call that crenation. Now, osmosis is the movement of water to balance concentration differences between those compartments. Now, the excess salt in the extracellular or outside space leads to water moving out of the intracellular compartments to balance the concentration. And too much water entering the intracellular or inside space of the cell can cause the cell to burst. You also call that lysis. Now, there are three tonicity, and that can cause water imbalances. The first one is hypertonicity. It refers to the extracellular space having higher concentration compared to the outside. The result of this is for water to have the tendency to move out of the cell, causing to shrink. Now, the concentration balance is very important for biological functions, and therefore, your cell needs to achieve isotonicity, which refers to the compartments with equal concentration on the outside and the inside of the cell. And lastly, for hypotonicity, this refers to the extracellular space having lower concentration, and so the tendency is for water to move into the cell, causing the cell to swell. Let's take a look at the diagram for each of the tonicity up close. For hypertonic environment, the tendency of the water is to move out of the cell, causing the cells to shrink. For isotonic concentration, if you notice, there's an equal amount of water in and outside of the cell. And for the hypotonic, if you notice, water rushes inside the cell, causing the cell to swell. The cell is affected by gravity, air pressure, and other physical factors contributing to your osmotic pressure. Differences in concentrations between intracellular and extracellular compartments create osmotic pressure. And osmotic pressure can lead to volume changes in compartment, swelling, or shrinkage. Now, for our lab today, you guys will be observing and exploring the osmosis model system. Now, there are three procedures that you're going to follow. The first one is procedure one, that has to do with exploring osmosis when two solutions separated by a semi-permeable membrane are identical. The second one is exploring the osmosis when different solutions are separated by a semi-permeable membrane. As you can see on the right-hand side, if you notice, there's a quite faint color. The first one is both water. The second one is water, and the other one is a test solution, which is guanine. And for the third procedure, you're going to take a look at cytochrome with unknown concentration compared to the water. So you're going to explore in the procedure three when different solutions are separated by a semi-permeable membrane. And this will also allow you to determine the concentration of this test solution. Now, please make sure that you follow all the procedures that you need as stated in our tabletop science platform. And if you have any questions, please let me know, and I hope this helps. Bye.